I'm Stephen Guest, and I'm the Director of Medical Consulting Services with Baxter Healthcare Corporation in Deerfield, Illinois. This presentation will be entitled Extraneal Icodextrin PD Solution Mechanisms of Action and Clinical Studies. First, I want to have disclaimers. The program is sponsored by Baxter Healthcare Corporation. I'm an employee at Baxter, as mentioned, as Director of Medical Consultancy. The objectives of the presentation were to go through these chapters, 7 in total. The first chapter will review the composition of extraneal icodextrin PD solution. Then we'll move to the second chapter, review the mechanisms of action of extraneal. The third chapter will be a deeper dive into peritoneal aquaporins. Chapter 4 will be free water ultrafiltration. And then our fifth chapter will be the discussion of crystalloid versus colloidal osmotic pressure. And Chapter 6 will be into the literature. Let's look at randomized controlled trials with extraneal. And then 7 will review some important safety information. First of all, though, I do want to start with that safety information a bit because extraneal has an important set of risk information. And this is a black box warning that you can see from the FDA, and you'll see along with this chapter, this particular slide, that extraneal is indicated for the single daily exchange. That exchange can be 8 to 16 hours. It can be used in CAPD regimens or the APD regimens, but it's for a single exchange, the long dwell. Extraneal is also indicated to improve, compared to 4.25% dextrose, the long dwell ultrafiltration and clearance of urea and creatinine in patients who are high and high average transporters, as defined by the PET test. But below that first paragraph, there is that black box warning, and it addresses a very important drug-device interaction. And the issue is many of the point-of-care glucose testing devices are themselves not specific for glucose. They can detect maltose in the blood and read that as glucose. In other words, give a false reading of glucose in patients that might have maltose in the plasma. So the black box warning refers to the type of devices where we would have this concern. And those are the devices that use certain reagents in the device, which this material here refers to. And those are devices that use reagents such as GDH-PQQ, which is a type of chemical reagent that the device uses to detect glucose. Those devices would be contraindicated, those with GDH-PQQ as a reagent. Also those that are GDO-based, and I'll let you read the full name for these. And then finally the GDH-FAD-based monitors. So at the bottom right you see a website, www.glucosesafety.com. That is the website that would list the monitors and what their reagents are, so that we would look at that website and see, are these reagents compatible with extraneal? And that's something that we would really have to check, so there's not this drug-device interaction. And the issue is if the patient is on extraneal and the device was detecting maltose, it would be a spurious elevated glucose level, where there could be excessive insulin administered, or the patient may actually be hypoglycemic, but the monitor is showing an acceptable level of glucose because it's detecting maltose. So this really is a serious interaction that we would have to be aware of. The bottom line is that patients that are prescribed extraneal, the clinicians need to make sure that the glucose testing devices in the home and in the clinic, as well as the hospital that the patient would be treated, would be compatible with extraneal. And fortunately, most glucose monitors are compatible, but it's still the obligation that we check this site and make sure these glucose testing devices are the correct ones to be used in patients that are on extraneal. So that is important safety information. It will be very important to review that on your own. Now there are some listed other risk informations which you can see on this slide, and just very high level. that it's contraindicated for any patients that have a cornstarch allergy. And we'll explain why. It's derived from cornstarch. It's not for IV use. It's for intraperitoneal use. Patients that have been on Dianyl or Extraneal have developed EPS, encapsulating peritoneal sclerosis. So that's a risk information. Patients have had some hypersensitivity reactions with Extraneal, usually involving the skin, such as you see on the slide. Toxic epidermal necrolysis, some angioedema, some serum sickness, rarely described, erythema multiforme, some vasculitis. So there's been some rare but serious hypersensitivity reactions to the product. The overinfusion of Extraneal could lead to overfill in peritoneal dialysis with increased abdominal pressure and a feeling of fullness. Patients with insulin-dependent diabetes may require an adjustment of their insulin. Extraneal is a non-dextrose product, so it may require adjustments in their diabetic regimen. Patients may have changes in protein removal or fluid and electrolyte changes during dialysis with Extraneal. And it may be required to intervene on any change in fluid and electrolytes. Finally, we look at some rare events that were reported in clinical trials, less than 10% of patients in clinical trials, but there were other rare occurrences such as upper respiratory tract infections, peritonitis, hypertension, some rashes. So again, this is information that's important to know about the safety of the product, and please review this. Now in Chapter 1, we'd like to review the composition of Extraneal icodextrin solution. It is derived originally from cornstarch. The cornstarch is subjected to enzymatic hydrolysis, which begins chopping down this large starch molecule into smaller fragments, which have been deemed maltose dextrin fragments. And then that is poured through a filtering membrane that further selects a fraction of a certain size, and that size is called icodextrin. And this is a slide that just shows the different molecular weights of that little fraction. It is a mixture of these different sizes of little chopped up oligopolysaccharides. The oligopolysaccharides are actually links of dextrose, of glucose. So we have glucose chains joined together in the 1 to 4 lineage, 1 to 4. And they form a large chain. And so these oligopolysaccharides are links of chains of joined glucose molecules at the 1 to 4 carbon. And so they're really large polyglucose molecules of varying lengths. I've had a picture made of what it appears. It's just a large, bulky molecule of these 1 to 4 linkages of the glucose molecule. And that is icodextrin, this larger polyglucose. But it's not glucose, it's a polyglucose. In this chapter, we'll review the mechanism of action of Extraneal. So Extraneal, as discussed, is a larger, bulky molecule, and when infused into the peritoneal cavity, is a large molecule that exerts what's called a colloidal osmotic pressure, meaning it has the ability to attract water toward it. So it might be even described to the lay person as a magnet that can pull things toward it, fluid toward it. So this large molecule intraperitoneally will draw fluid out of the plasma toward it. That's the purpose. That's one of the functions of a colloid. And I think you can see in that picture it's a large yellow molecule, really too large to be absorbed across the capillary, and it just is attracting water towards it. That picture shows that it is too large to be absorbed across the capillary, but it is absorbed eventually. The mechanism is believed to be through the lymphatic channels, particularly up under the diaphragm. We have lymphatic stoma that are large enough to allow icodextrin molecule to pass into the lymphatics, where it goes in the lymphatics up eventually into the thoracic duct and enter the superior vena cava. And at that point, icodextrin does reach the blood supply. But it's not absorbed directly through the peritoneal capillary. It's a more circuitous route through the lymphatic system. Now, this shows the dialysate icodextrin on the left versus time, and you can see what looks like a very linear, slow absorption of icodextrin. It begins to slowly disappear from the peritoneal cavity in a very predictable, linear way as it reaches the lymphatics and eventually the plasma. So this slide, quite important, and it will show the entire metabolic fate of extraneal. It starts in the peritoneal cavity, infused in the cavity. It eventually, through the lymphatics, reaches the intravascular component. And when it reaches the intravascular component, being a starch, it is subjected to pancreatic amylase. And pancreatic amylase starts to do its digestion of starches and breaks these large glucose chains into smaller and smaller fragments. The ultimate fragment is a 2-glucose chain called maltose. So in the intravascular department, we have amylase digestion of icodextrin up to maltose. Maltose, being a 2-dextrose chain so small, it can at that point migrate into the intracellular compartment, where it's exposed at that point to intracellular maltase, which breaks the 2 glucose molecules apart into individual glucoses, which are used for cellular energy within the cell. So at no point can you see glucose in the intravascular space. It is eventually turned to glucose only within the intracellular compartment, where it's used for cellular energy. So it is considered a non-glucose dialysate solution because of this unusual metabolic fate. This picture shows on the left the plasma levels of these different chains as amylase starts to do its digestion, and you do see different levels of change. The smallest one is the maltose, but then you have maltose trios, maltose tetros, and you can see different chains all undergoing amylase digestion. If that continued, eventually it would just go to maltose. But at that snapshot in time, you can see the different digestive properties, digestive components of the original molecule. Now, on the left, we have plasma insulin levels. On the right is plasma glucose levels. And you can see how stable. This is not a dextrose-based solution, so there's not changes in plasma glucose as mentioned. And if there's not a change in plasma glucose, there's not a change in plasma insulin. So you see the stability in insulin and glucose levels because of it, as we mentioned, the only glucose that appears is within the cell. So on the left is our extraneal solution. On the right is our dianeal solution. And besides the differences between dextrose and icodextrin, you can see the similarities. Similar sodiums, chlorides, calcium concentrations. There's a magnesium. A lactate is the buffer on both of these solutions. They're both kept at a lower pH. So high-level review of the icodextrin pharmacokinetics. The absorption is kind of convective absorption up through the lymphatics. This says up to 60 grams absorbed in 12 hours. Metabolized in the blood plasma by amylase, eventually to maltose. Very excreted eventually by renal or dialytic clearance. Plasma levels reached steady state within a week. They didn't see accumulation of maltose in tissues. It was a steady state. Again, stable over long-term therapy. And if extraneal is discontinued, it's important to remember that those maltose and some of those smaller digestive properties can remain in the plasma up to 2 weeks after stopping extraneal. So those glucose testing devices may still be a problem and cross-react with maltose up to 2 weeks after stopping extraneal. So we have to keep that in mind. And then just a comment at the bottom there that icodextrin has been not fully studied in certain subpopulations, but age, gender, race, for example. So here's our classic ultrafiltration profile of a 1.5% dextrose solution. All the different PET categories at the top. We have the the low transporters showing the most significant ultrafiltration. High transporters absorb dextrose rapidly, a less rigorous ultrafiltration profile. But all of the dextrose curves, as the audience is aware, they peak, plateau, and then we reabsorb that ultrafiltrate over time. That's just the nature of the dextrose-based solutions. 1.5%. When we move up in osmolality to a 2.5% and look at the different PET categories, we could get up to a 600 milliliter ultrafiltration with a low average. High transporters may give us 200, a bit more ultrafiltration, but again, over time there's a peak ultrafiltration, and as the dextrose continues to be reabsorbed, we lose that ultrafiltration, we reabsorb it, and as you can see in the high and high average transporters, we could actually begin to absorb the bag itself of the dialysis solution. The 4.25, our strongest dextrose solution, will again give a more sustained ultrafiltration, but it too will peak, and then we reabsorb that ultrafiltration back over time. Those are the dianyl classic ultrafiltration curves. Superimpose the extraneal curve. Extraneal is a colloid. It sits in the peritoneal cavity. It attracts fluid toward it. It's only absorbed in a very linear fashion, so you get a very consistent ultrafiltration. Some people would refer to that as just a constant tortoise in the hair or whatever, but it's a constant ultrafiltration that you see profiled in that particular slide. The other issue with icodextrin is it works across categories. The PET categories were really referring originally more to clearance and dextrose ultrafiltration. The icodextrin, being a colloid pulling fluid by a different mechanism, it's not based on PET categories. It's a different way of pulling fluid into the peritoneal cavity. So the slide just indicates that the ultrafiltration profile is fairly consistent across all different PET categories. Now, this is a slide showing how the osmotic gradient is maintained over the dwell time. And let's look at the green first. With dextrose-based solutions, dextrose is absorbed more rapidly. You can see the loss in that concentration of dextrose from the intraperitoneal cavity, as opposed to extraneal, which is not as readily absorbed. And so over the dwell time, you have more of the active reagent within the peritoneal cavity, explaining why we have this more sustained ultrafiltration. Again, superimposing dextrose-based solutions versus extraneal, it's a different ultrafiltration profile. In this chapter, we will review peritoneal aquaporins. The peritoneal capillaries have been modeled as having 3 pores, the so-called 3-pore model. The peritoneal capillaries were modeled with the understanding that there must be larger pores, but not many pores, because there is a small amount of protein that appears to move from the plasma into the dialysate. And protein being a larger molecule, it was implied there are larger pores, but not many. They're modeled that there must be smaller pores, and many of those, because the smaller pore is the pore that is believed the creatinine and urea and smaller molecules are transported across. The third pore identified was a pure water pore, a pure aquaporin. So these are the classic pores within the peritoneal capillary that allow for substances to move from the plasma into the dialysate. The aquaporins, as pictured here, are, as the name implies, water pores, water channels. And when we put a substance such as dextrose-based solutions on one side of that capillary, in the peritoneal, peritoneal cavity, the dianyl has a higher osmolality than the blood. So now the peritoneal capillary is perceiving 2 different osmolalities on different sides of the capillary. And membranes tend to not want to tolerate differences in osmolality across the membrane. So the aquaporin allows for water movement to move from the osmolality of 280 toward the higher osmolality to try to dilute that osmolality down. Preferably, we want an equal osmolality on both sides of a membrane. So again, the dianyl is infused into the peritoneal cavity. We have a perceived difference in osmolality across that capillary. Aquaporins allow water movement to try to dilute that osmolality down, but that is our ultrafiltration. That is a water ultrafiltration. One of the levels of evidence to suggest that there were these aquaporins were when dextrose-based solutions were infused, but then drained out early in the dwell, let's say 1 and 2 hours, and researchers checked the dialysate sodium concentration. So the starting dialysate sodium concentration is 132. So when they drained a 1.5, 2.5, and 4.25%, as you can see in this slide, and rechecked the dialysate sodium concentration, well, gosh, with the 4.25%, you can see the dialysate sodium concentration might have been detected at 124. So they determined, wait, if the dialysate sodium concentration is diluting down from 132 into the 124 range, the only way that it could occur is pure water must be moving from the body into the dialysate to dilute the dialysate sodium. And sure enough, they determined that there were aquaporins and that there is this water movement. And so again, the evidence was that the capillaries have pure water channels, aquaporins. In this chapter, I'd like to review that concept, the concept of free water ultrafiltration. So pictured here is the peritoneal capillary aquaporin. And water moves across that aquaporin in response to differences in osmolality. Water moves across, as you can see in those arrows, but the sodium molecule, which is the white stars in this pictorial, cannot move across the aquaporin. Let's look at another picture. I see water moving across that cell in blue, and the sodium cannot pass. That is called sodium sieving. Aquaporins allowed pure water to move, but that sodium cannot pass. It is sieved. It's sieved at the level of the peritoneal capillary aquaporin. Now look at the slide there at the bottom. 48%, 44 to 48% of the ultrafiltration during peritoneal dialysis is water. It is aquaporin-mediated water ultrafiltration. The sodium that's sieved, if I did another exchange too quickly, sodium would build up. It's sieved. We have to slow solution exchanges down to the point that sodium has time to diffuse through the small pores. It's not allowed to pass through the aquaporin. It slowly diffuses through the small pores. And that's part of the reason you don't see patients on a cycler with 10 exchanges. There would be water movement without the sodium. Water without sodium. Patients early in the literature became thirsty, hypernatremic. So we learned that we have to have sufficient dwell times to allow that sieved sodium time to diffuse through the small pores. Again, top line PD with glucose-based solutions. We see water movement across the aquaporin. The sodium is sieved. It's retained in the plasma. But then that sodium and water can move through the small pore, a bit to the large pore even. And then PD with icodextrin will have a different mechanism. You see a different pattern there, which we will discuss. In this chapter, I would like to review crystalloid versus colloidal osmotic pressure. In this image, we have a picture of a dextrose-based solution and the fluid movement. We have, as mentioned, up to 44 or 48% of our ultrafiltration is across the aquaporins. The remainder of the ultrafiltration is largely across the small pores. So again, dextrose-based solutions involve aquaporin-mediated ultrafiltration and small pore-mediated ultrafiltration. This is the picture of icodextrin-based ultrafiltration pattern. We have only minimal aquaporin movement of water, 4%. The the predominance of the ultrafiltration with icodextrin is across the small pores. This is a fundamental difference in the types of solutions. Dextrose-based solutions have substantial water-only ultrafiltration across aquaporins. Extraneal, being a colloid, does not induce a water-only ultrafiltration. It pulls a fluid across the small pores. I went over this slide fairly rapidly in previous chapters on purpose. But if you look again at the left side of the slide, the extraneal concentration, and then to the right, the dianeal. We did talk about the differences in dextrose, icodextrin, sodium, the same chloride, lactate, but go look at that osmolality. Extraneal has an osmolality in the 280s, the same as plasma. So extraneal is isoosmotic. Therefore, when it is infused into the peritoneal capillaries, into the peritoneal cavity, the capillaries do not perceive a difference in osmolality. The plasma's 280 milliosmoles, the extraneal 280 milliosmoles. There's no perceived difference in osmolality, so no perceived need to move water across the aquaporins. No water-mediated ultrafiltration. The ultrafiltration is largely salt and water coming across the small pores. And I'll just have this picture, we have this drawing to just illustrate that in another way. On the left we see dextrose or crystalloid-based ultrafiltration, which is 48% pure water. And then the remaining 55% would be salt and water. When we look to the right with the icodextrin or extraneal profile, very minimal water-only ultrafiltration and a substantial ultrafiltration that is salt and water. And so I think that an understanding of what we need to truly remove in that ultrafiltration, is it water or is it salt and water? I think you can understand that these are fundamentally different solutions in the type of ultrafiltration that they create. All right, a couple of key concepts. Number one, what is the difference between dianeal dextrose solutions and extraneal icodextrin solution on peritoneal capillary aquaporins? Well, dianeal dextrose solutions are crystalloid. They have a higher osmotic pressure, and they will induce pure water movement across the peritoneal capillary in response to those differences in osmolality that the peritoneal capillary perceives. Extraneal is isoosmotic. There is no perceived difference in osmotic pressures, so there is no perceived need for water movement to equalize or alter any osmotic pressures. Number two, what is the difference between crystalloid osmotic properties and colloidal osmotic properties on peritoneal aquaporin function? I think we just covered that. Icodextrin extraneal is isoosmotic. Peritoneal aquaporins play a minor role, if any, in that ultrafiltration that is induced. It is a salt and water-rich ultrafiltration. In this chapter, we will review some of the published studies regarding extraneal use. There were early randomized trials. You can see on the left the mystery trial. The trial was 1994. And that was an early trial of 209 CAPD patients over 6 months, randomized to dextrose versus extraneal to try to understand basic safety and efficacy. And they concluded in the study that icodextrin could be used in the long dwell as a replacement for dextrose solutions. That's 1994. Then it moves to 2002, where 2 studies came out in the literature. The Plum study was now APD patients, 39 patients. They were randomized to extraneal versus 2.5% dextrose for the long dwell. And the conclusion of that early study was that there was an increased ultrafiltration with the icodextrin. Now the Wolfson study came out 2002, was actually 2 buried randomized trials published together in the article. One was a shorter-term 4-week study that was again looking at safety and efficacy, followed by a 52-week long-term study, the longest to date, again looking at longer-term safety and efficacy, and they concluded that the ultrafiltration and clearances increased in the group with icodextrin. Very early studies, decades ago. Now, we move to the 4th randomized trial. This is Dr. Davies. This was a study of 6 months, and what they were looking at was the body weight changes during icodextrin-based long-dwell versus dextrose. And as you see in the left panel, the dextrose group over the 6-month trial gained in body weight. The icodextrin group showed a contraction in body weight. So the next thing they did in the right panels is the researchers used bioelectric impedance to say, well, if the weight is being decreasing with icodextrin, what's changing in the weight? And they saw a contraction, as you can see in the top panel, they saw a contraction in the icodextrin group in total body water. So then they asked the question, well, of the total body water, what is contracting, intracellular versus extracellular water, again based on bioelectric impedance. And as you can see, they confirmed that the contraction is in the extracellular fluid, which is one of the goals of dialysis. And this is an important study showing there is a contraction in body weight, and it's from the extracellular space. Now, many of you in the audience may be familiar with this trial because this was a randomized controlled trial based in the United States, and this was a head-to-head comparison of 4.25%, our strongest dextrose solution with Extraneal. The researchers looked at some of the metabolic issues aside from ultrafiltration and looked at carbohydrate absorption and calories that are absorbed. On the top row in purple, the 4.25%, you can see the carbohydrate absorption, the caloric absorption, and then a term called ultrafiltration efficiency. And what they meant by that term is how much fluid can be removed from a patient at a metabolic cost. In other words, how much fluid can be removed in ultrafiltration for dextrose or carbohydrate absorption, that metabolic cost for your ultrafiltration. And they term that ultrafiltration efficiency, the mils of UF for the gram of carbohydrate absorbed. So you see the dextrose panel there. Look right under that with light blue. The Extraneal carbohydrate absorption was decreased. The Extraneal caloric gain was less. And the ultrafiltration efficiency, the ultrafiltration per gram of carbohydrate absorbed was double. Then they looked at ultrafiltration. And again, this is head-to-head, the strongest dextrose solution, 4.25% with Extraneal. And within the first week of the randomization, they detected a superior ultrafiltration that sustained in the second week of the study. And that, again, head-to-head randomized trial. This is not a randomized trial, but I do want to show another important Extraneal trial. And it was a study by Dr. Davies looking at longitudinal peritoneal membrane changes. And they looked at the patient's D over P creatinine and some of the issues related to membrane properties over time. And in the open squares, which were dextrose, you can see that patients over time move toward higher transport status. This has been described in the literature. Patients exposed to higher dextrose concentrations in the dialysate over time have been described to develop a neovascularization in the peritoneal cavity that translates into a higher transport status, faster transport. So you can see that migration toward higher transport status in the squares. The dark circles is the Extraneal group. And over the study, in this published study, over time, the Extraneal group showed membrane preservation. There was not this migration toward higher transport status. Again, it's a non-dextrose solution. So that was an important study that gives some information about internally the effect on the membrane. Back to our randomized control trials. Now this is number 6. This was a randomized control trial, icodextrin versus a 2.5% solution. And this is the study, if you were wanting that evidence, that looked at icodextrin with all different types of transport properties. And again, this is Clinical Journal of the American Society of Nephrology. The prior publication was in a prestigious journal. So this group, head-to-head, 2.5 randomized control trial. It was a 2-week and a 4-week. And you can see on the left panel that within the 2-week measurement, the group with icodextrin had the higher ultrafiltration in the dark squares. That was maintained in the 4 weeks of the study. On the right panel, you can see the ultrafiltration based on PET category, and the low average, high average, and high transport patients all show statistically significant ultrafiltration with icodextrin. So again, that was a study fairly recently, 2009, showing extraneal by PET categories. Now we move to a 2015 randomized trial. And this was called the STARCH trial, and this was looking at icodextrin use in non-diabetic patients who were undergoing APD. Now patients on peritoneal dialysis have been described as having a state of insulin resistance and can develop insulin resistance possibly due to the exposure, constant exposure of dextrose absorption over time, this potential to develop insulin resistance. And so this measured the degree of insulin resistance in patients who were non-diabetic but maybe pre-diabetic, insulin resistant, icodextrin versus dextrose solutions. They use what's called the HOMA index. And the HOMA index, you can look that up separately, but it involves measuring the pancreatic amylase levels, and it measures serum insulin levels, and it's an index calculation that can calculate the degree of insulin resistance in the body. And on the left panel, you can see that they noticed that there was a bit higher HOMA index with the dextrose group. In other words, more insulin resistance in the dextrose group compared to icodextrin. And on the right, just again, the change in the HOMA index over the 45 and then 90 days of the study. The dextrose group showed an increase when you got to the 90-day parameter. And extraneal being non-dextrose, there was a contraction in that insulin resistance index. That's a 2015 study. So again, there are other randomized trials that we just don't have time to discuss, and I looked at all of them. The gold standard in the literature is a randomized trial, and there are 11 randomized trials on extraneal use. In Chapter 1, we reviewed important risk information associated with extraneal. In Chapter 7, we're going to expand on that safety information. So in the top paragraph, again, extraneal is indicated for a single daily exchange, the long dwell of 8 to 16 hours, in patients on CAPD or APD. And it is indicated to improve ultrafiltration and clearances compared to 4.25% solution in patients that are high average and high transporters. The box that you see on the slide is that important drug-device interaction, which we spoke in Chapter 1 and we'll cover in a slide. These are the different other important risk information. It's important to review these for each caregiver that's employing extraneal. We have extraneal contraindicated in patients who have a cornstarch allergy, as mentioned. We have extraneal indications only for intraperitoneal use, not intravenous use. Patients have developed EPS with extraneal as well as dextrose-based solutions. There's been serious hypersensitivity reactions, usually involving the skin, involved with patients exposed to extraneal. Patients that are over-infused with extraneal could develop an abdominal fullness and overfill, as they could with dextrose-based solutions. Patients who are diabetic and taking insulin may need alterations in their insulin dosing, as Extraneal is considered a non-dextrose solution. And then patients that are on Extraneal will have possibly changes in fluid and electrolytes that would need to be monitored and treated as indicated. And the last bullet here is showing the other various adverse events that appeared in these early trials that we discussed, and they include hypertension, some respiratory infections, peritonitis, and rash. So the glucose monitor interaction with Extraneal icodextrin, again, this is a drug device warning by the FDA in regards to certain point-of-care glucose testing devices. Those devices may not be specific for glucose. As the first bullet describes, maltose may interfere with some of these blood glucose devices. And the devices that are of concern are those using the second bullet, largely GDH-PQQ as a reagent. But you can see in the last bullet it's not just GDH-PQ, there's a GDO-based reagent as well as a GDH-FAD-based method. Those three are contraindicated in glucose devices in patients with Extraneal. The patients have a wallet card that is a very prominent warning about this device-drug interaction. Again, glucosesafety.com is an available resource listing all point-of-care devices to tell the reagent that's used and whether it's compatible with Extraneal. We have several other types of reactions in those trials I mentioned, the various common ones. We had peritonitis reported in early trials. There was upper respiratory tract in icodextrin as well as control groups. You can see the differences in these adverse events, dextrose versus icodextrin. The most significant one it really is the rash and these skin hypersensitivities. An interesting nuance, if you use that word with Extraneal, is that the Extraneal solution can interfere with amylase testing. And that is because, as we mentioned, when Extraneal moves through the lymphatics and appears into the plasma space, pancreatic amylase begins digesting the oligopolysaccharides. The way that the laboratory measures amylase is to take a set part of the blood and test the blood against the reagent in the laboratory and to test amylase digestion of this reagent. But because Extraneal is in the patient and the amylase is over here distracted by the Extraneal, there's not the same amylase availability to react to the reagent in the laboratory. So the laboratory test detects a low serum amylase value. It's really a kind of a competitive inhibition. So that nuance means that if a patient is suspected of having pancreatitis, the patient should be tested with lipase. The amylase will be read as spuriously low. It may mask a pancreatitis case. So patients on Extraneal that are suspected of having pancreatitis should be tested with serum lipase. There's a decrease in serum sodium slightly with Extraneal use. And it's felt to be that this colloid is pulling fluid, that it can create a fluid movement from intracellular to the extracellular space, and there can be a dilutional hyponatremia. It's believed to be a dilutional hyponatremia. The other factor is that these metabolites that are absorbed into the blood plasma space themselves are osmotically active. They may also attract fluid from the intracellular. The net effect of this fluid movement is the description of a dilutional hyponatremia, somewhat similar to hyperglycemia. A couple of other important clinical messages in conclusion now. Extraneal is indicated to improve long-dwell ultrafiltration compared to the 4.25%, and is particularly in high and high average transporters. It increases the long-dwell clearance of creatinine and urea compared to 4.25, particularly in those high and high average transporters. Icodextrin improves the fluid status compared to a 2.5% dextrose solution in high and high average transporters treated with CAPD or APD. And again, these are the clinical messages that have come out of the 11 randomized trials. Other clinical messages: Extraneal reduces the long-dwell carbohydrate absorption. That was the Finkelstein study, compared with 4.25% dextrose in high and high average transporters treated with APD. And again, you cannot repeat the importance enough. Only glucose-specific monitors and test strips should be used to measure the blood glucose level in PD patients with Extraneal because of this interaction with the drug and the device. Additional information on the product or therapy questions in regard to Extraneal can be directed toward Baxter's Medical Affairs. There's some clinical helpline information here, both telephone and fax, and hopefully you would take advantage of this information if there are any additional questions on Extraneal. Again, further information is at theglucosesafety.com in terms of the point-of-care monitors. And you could also contact your Baxter clinical educator for additional information on Extraneal. Thank you for viewing today's presentation. I hope this information was of interest to you and expanded your knowledge of Extraneal.